Okay, so very good afternoon, all of you. And today's 23rd. We are slowly moving towards the end of this course, and I'm very happy that you could make it for today since we have changed the timings of the class yesterday. So thank you for joining. And we have with us Dr. Manash Chatterjee, and I would now request Professor Sujata Bhargava to kindly introduce our speaker for the day.
Good afternoon, everybody. I'm privileged to introduce you to today's speaker, Professor Manash Chatterjee. He is a plant biotech scientist and founder director of a company called BenchBio, which is located at Vapi in Gujarat. His specialization is in applying breeding techniques to create new crop varieties having novel traits. He focuses on a technique called tilling, about which he will talk today. He has created the world's first non-GMO papaya ring spot virus resistant papaya, which he has patented. Regarding his educational background, he studied at the GB Pant University and IRI, after which he did his PhD at John Innes Center at UK. He did his postdoc work at Salk Institute in San Diego, USA. He has also been in teaching positions. For example, he's working as an adjunct faculty position at the National University of Ireland. And he's a senior member at the NN Ed Pro Global Center for Health and Nutrition. I think it's a think tank of the UK, which deals with new agricultural technologies, amongst other things. He is also a member of the advisory committee of Club Demeter of Paris, which is a consortium of agro-food companies as well as higher education institutes. So, with a very impressive journey which he has made so far, I'm sure all of you are eager to hear him talk about this technique of tilling. And I request Dr. Manash Chatterjee to start his talk.
Thank you very much, Dr. Sujata. First of all, I would like to thank Dr. Sujata and organizers for inviting me. It's an honor and a pleasure to tell you what my company has been doing. So, without any delay, I would like to start. So, welcome everyone. Good afternoon. So, the talk is about tilling, which is a non-GM method to improve crops and industrial perspective. So this company was set up in 2007. It's a non-GM technology which was licensed from France. It was a patented technology. So there was an enzyme which was discovered by one of my collaborators, who is deputy director of this lab called IPS2 as part of INRA. So INRA is actually equivalent to ICAR of India. So the unique selling proposition of our company is that we are working and creating non-GMO traits. We are creating traits in crops using a non-GMO method. So it's a it's a science-based plant biotech company focused on creating new traits in crops using tilling. So we are focusing on discovering induced alleles from plants. So which is, a mutant allele discovery platform we have called M-tilling or tilling. Focused on discovering natural alleles from plants or natural allele discovery platform or eco-tilling. And we are focused on genes for agriculture and consumer value. And we are working on food, feed, as well as non-food crops. So, this is the beauty of this technology, that once you have it, you can actually apply it to any crop produced from seeds. So the company is located in Vapi, which is the western part of India, Gujarat, Maharashtra border, 3 hours drive from Mumbai. If you happen to pass on that side or you want to visit, you send me a mail, you're welcome. So since this talk kind of is semi-academic, so I thought, maybe I'll put some 2 or 3 slides explaining the science behind it, that this technology, many people don't believe in this technology. They think it's like shooting in the dark, but I can convince you hopefully today that that's not the case. So basically, EMS leads to 100% GC to AT transition. We know that. Let me hide this. You're able to see my screen? Okay. 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 So, sorry, I do not want to touch anything right now. So, okay, so EMS leads to 100% GC to AT. So the point of this slide is that can saturation mutagenesis be used to improve crops? What I mean by that is, can you really mutate every gene in the genome? Okay, and I will hopefully give you evidence that you can. So this is a slide I borrowed from my collaborator, Dr. Ben Haman, who invented this tilling technology in Europe. So what he did was he actually tilled 5 different genes from a population of Arabidopsis of 125,000. And he's showing that you get different kinds of mutations. So the conclusion was that you usually get 5% truncations, 64% missense, and 31% silent mutations. And then the interesting part, if you look at the amino acid chain, you can see that, about 75, even up to 80% of your amino acid can be mutated. So basically, this is like site-directed mutagenesis, if you can do it very well. And so this means that if you want to hit every gene, maybe if you have the size of the population a bit large, like in tomato, we calculated approximately 100,000 should work. And so we have also been working on these large populations in various crops. And one of the advantages of being in India is that, we can grow large populations in India. We can grow any crops we want to grow because of the climate. So this is what we are taking advantage of. This is another slide to just remind you people how breeding is done, in the sense that, let's say I'm a breeder and I want to breed a trait. So traditionally what you do, you actually, maybe there's nothing in your germplasm collection. So what you do, you try to find, is there a germplasm available in database or some seed bank, and you like AVRDC, you as a breeder would like to then write to them, they send you the seeds, you start crossing with that. It can take 3 to 5 years, even more. So, but you have to understand that competition between breeders is not anymore limited by access to germplasms, which means that if you as a company can get the germplasm, another company can also get that germplasm, because these are public bodies. They share the germplasm with, as many people as they want. So can you do something with the same material and create something which is different to what everyone has? And I think this is the logic of BenchBio, that if you have tools for allele engineering and the knowledge of the genes for the trait, you can actually modify and maybe improve and enhance the same germplasm which you have obtained. Okay, and so then differentiate yourself from other seed companies. And this is where we have been helping seed companies worldwide actually to improve their material and create some new traits, solve their problems. So BenchBio platform basically comprises of high quality mutant population creation technology. This is very important. Many tilling labs suffer because they are not able to generate this high quality mutant population. Our mutant populations are very high quality. We get about 1 mutation in 200 kb in most crops. And as a result, we end up getting like 25 mutants per gene. In flax, we got 1 mutation every 42 kb. We were getting 50 mutants per gene. So this was an academic collaboration. So that was fantastic for them to find functions of genes. So yeah, so we have in our platform high quality mutant population creation technology. Once you have this, You can do reverse genetics or killing. So you know the gene you want to screen for it, and you get a trait. So herbicide tolerance is an example. On the right hand side, one of the beauty of tilling is also that you can do forward genetics, especially for traits for which genes are not known. You can, screen by old-fashioned method, plant your population, screen in hotspots or some traits we're interested in, and then you get the phenotype. And if the reference genome is available, even if it's not available, these days the cost of sequencing is not very high. You can sequence it, and you can then, using bioinformatics, map it, and then you can, find the mutant, clone the gene, and you have a marker anyway, so you can start building program. So that's the power of this. And the other important which you need for success of tilling is candidate genes. So I can tell you that when I was starting my company, people told me, where are the genes? Where are the genes? It will not work. It will not work. And today, there's so many genes. I'm overwhelmed with genes. You type something, you find something. And the other thing is that many of the patents on GM traits, transgenic method of creation something, those usually don't cover those genes. So they will have a specific, protection like method to create transgene and all that. And so any antisense, RNAi and all that, they are candidates for, tilling. Okay, and this we are taking advantage of. So what we are doing in tilling is basically from sequence to phenotype, we are engineering alleles in crops, and we are inducing these alleles by mutagenesis. Okay, so little bit of history. So it's a novel technology that allows rapid selection of mutants from any gene in any organism. The initial development and testing was done in Arabidopsis. It was actually a technology created by a PhD student in 2000 in Fred Hutchinson Cancer Center in the US. Tilling now applied to all crops and organisms such as mouse, fish, flies, and worms. Of course, it's a functional genomics tool. Basically, you are creating a library of mutations and you are screening that for functions as well as trait development. Public and private labs worldwide now engaged in tilling. You Google tilling crop, you will find there are labs all around the world doing it because it's non-GMO. So that is preferred especially by countries outside of the US. So in a nutshell, what we're doing is that we are taking advantage of large amounts of sequence information available in public databases. So, what I mean by that is the sequence information in CBI, you need a bioinformatician, a computer, internet connection, and you start mining and, getting list of traits you can create by tilling. So here is how the platform looks like. So basically, this is the enzyme-based tilling. So basically, we call, just to make you understand, so we call untreated seeds M0. So let's say we start a project and we are given a bag of seed by a company to create something. So we receive the seed in BenchBio, we call it M0. Okay, we treat with EMS, we call these seeds M1. We plant M1 seed, we get M1 plant. Okay, in each M1 plant, one copy of the gene is mutated. So you collect from each plant very carefully, without any mixing, bags of seeds. So let's say plant number 1, you collect 100 seeds from each plant. These plants are sick plants, they're carrying mutations, so you have to take extra care to grow them very carefully. Okay, and then what you do, let's say each packet has about 100 seed. So you keep 90 seeds in the seed store. You take 10 plants and grow them, 10 M2 seed families, and grow them in a pot. So we'll have maybe 10 seeds in each pot, plant number 1 to plant number 1,000 or 5,000. You then extract, you take leaf punches because they are segregated. Pooling. In the earlier method, it was 8-fold pooling. Then what you have to do is, a gene and the sequence is known. So let's say we want to make long shelf life tomato. The NOR gene is a very famous gene. It's out of patent. So basically you design primers to NOR and you start screening your DNA collection. These primers are IR dye labeled, so green and red. So they are detected by infrared in this machine. Uh, this enzyme, which was patented by Indra, it's a highly sensitive enzyme. It cuts single point mutations. So you do a PCR, you then subject this PCR to this enzyme. This enzyme cuts it, you run on this machine called Lycor. You get a green and red spot because of these iodide labeled here. And this means that these lines are carrying the mutation. You then send the DNA for sequencing. You confirm, yes, there is a mutation. And basically what we call is that we have created a prototype plant with a trait in mind. So let's say long shelf life tomato here. And then, of course, you have to do further work where you have to cross it and clean the background. And, so that's the breeding part you have to do. But this whole process, it takes about, I will say, 12 to 15 months. So let's say tomato, 6 months to grow 5,000 M1 plant, 3 months to extract DNA, and then basically another maybe 3 months it used to take us because these primers come from Europe. So it takes about 15 to 30 days sometimes. So yeah, but still, it's still, I will say, something which is working, something which is non-GMO, you will be able to actually commercialize it in the end, unlike GM thing. I'm not anti-GM. I used to work on GM crops. It's just that public doesn't want it. So we have to, so we were thinking of some alternative, and that's how this idea came. And the beauty, as I explained to you, that once you have this setup in your lab, you can process any plants which is produced from seed. Okay. You can also do vegetative plants. We are not doing it. There is a group in Vienna, in Austria. They are working on banana and many other crop with tilling. It involves tissue culture and all that, but we are focusing on seed-based production. Okay. So here is a newer application of tilling where basically because of Illumina, the power of tilling has become much, much higher in the sense that in this old method, we used to cut with enzyme and then you actually send it for, you detect and you send it for sequencing. Here what's happening is we are actually, you avoid this enzymatic step. You directly go and sequence and you get an Excel file with all the mutations. The beauty of also this is that you can do multiplexing, so you can have, libraries of genes, maybe, 10 or even more, and from 10 or 15 different crop species, put them in one tube, run Illumina, and then you analyze your data. So very fast, high throughput. And as I said, now I think this is taking about 12 months. And then, of course, evaluation and confirmation, maybe another 6 months. So by end of the second year, you have your mutants showing the trait, and then you start crossing, breeding, cleaning, because there are mutations in the background. And you can do speed breeding, and really by maybe third or fourth year, you could think of, having commercial variety. So advantages of tilling and end-tilling is that it's non-GMO. So, I live, I've been working in Europe, also in India. You can see transgenic, even though we as a scientists know it's a fantastic tool for some crops, it is not being accepted by consumers. And so there's a block, and I don't know whether this will become easy in the future. I hope so. It yields a spectrum of alleles, high throughput and relatively low cost of product, significantly less expensive than transgenic, no regulatory dossier you have to make, less regulatory hurdle to market, 3 to 4 years faster in some cases. So there have been examples, even in India where they have released varieties, by tilling method. So BenchBio is a company, what we do. So we are really a science-based company. So, but as a company, we have to generate income, to run the lab. So we have one, I call services, which is like, I call it dry cleaning, in the sense that companies tell us, do this, do that, and we just do that. So like they want us to analyze their germplasm, they want us to create populations in them. And then, and being in India, we are able to create large number of populations which they love. And so we are doing this. and we have been working on all kinds of crops, all of these categories. This is the part which is actually the key part of BenchBio, which I love in the sense that kind of like risk sharing and trying to solve the problem of companies. So we target specific traits and specific crops. We are writing sometimes grants between academic and us and all that. And so I'll show you examples of that. So our facilities, so, we are a small company and we have, glass houses to grow crops, air-conditioned glass house, non-air-conditioned glass house. We have land leased in Bangalore, in Kulu. We have a small molecular biology lab, so we have everything to do work. We have been now working for 14 years. So just to show you, when I say population creation, it really is a specialized activity. It is not an easy task because these plants are sick. So, but, we are able to grow. And as I explained to you that we are quite well known, especially in the European seed sector, that, our populations are very of high quality. So we are getting a lot of projects on that. So we have sunflower here. You can see here capsicum. Normally we have 5,000 minimum to 10,000, and then we also have worked on 50,000 tomato and other crops. So this is a tomato. When we're harvesting the seeds, we basically take a picture. If we send something unusual, so you can see that we get all kinds of things, and we have a database where we keep information about these lines. So tomato is our model crop in the sense that when I brought this technology from France, they were working on tomatoes, so I just copied what we're doing. So most of the work we have been working in-house is tomato, and of course to set up the platform. Once the platform was set up, we have been working on other crops as well. So this is sunflower, European sunflower population, big populations, about I think 25 to 30,000 M1 plants. Papaya, I'll show you an example of that. This is a very challenging project. I think I was able to do it because I was working with fantastic people and seed companies and team. Papaya segregates into male, female, and hermaphrodite. So we used marker to select hermaphrodite plants so that we only collect self seeds. So that was quite tedious and really challenging. So, basically, this just shows that we have created a platform of mutant libraries and various crops. So these crops lines belong to seed companies, so I'm not allowed to just distribute to anyone. Once the work is done, I return the seeds back to them. We have in-house, we have our own crop is tomato. So rest of the crops we're seeing is mainly crops which belong to other seed companies. So unable to share with anyone if they want to screen our line at the moment, but we are in the process of making a collection of our own. So again, as I explained to you that I, as a scientist, I publish, that's my marketing material to show that, we understand this area. And so this is the first paper we published, which was picked up by Science Daily. Anyway, so basically promote our work, as a top class, science company. So, so far I talked about population, things you need. So population, you need high quality. Then you need reverse genetics, Now I'm going to tell you about the forward genetics part as well, which we are doing where, for example, we grow M2 families, and we then have retired breeders or experts who are then given a list of traits and their screening. In this case, we don't know the genes. We will clone the gene afterwards. So here is an example where we found some mutants. So this is a mutant actually, which was identified by Nunums, and this mutation is called full flesh. This tomato doesn't have any water. And actually, this tomato of Nunums, one innovation, non-leaky tomato. So the burgers, in McDonald's, they don't become soggy. So there was an interest by some companies if we can find something similar. So we phenotypically, if we found tomatoes which were seedless, we don't know the gene. So we are actually now in collaboration with the French lab trying to clone this gene. And how do you do that? Because the tomato reference genome is known. So what you have to do is cross this with the original wild type, unmutated, you get F1, you get F2. F2 will again segregate, you again phenotype, and then you take pools of DNA from seedless, seeded, and you sequence it, and then because the reference genome is known, bioinformatically, you will be able to map the gene and have a marker and then finally clone the gene. Okay. So you can also do for disease resistance as well. We have some projects, but because it's a private project, so I'm unable to show that. But I can tell you that it's being used worldwide for traits for which genes are not yet identified. So to summarize, so we have a population, large populations. You do reverse genetics if you know the gene, like herbicide tolerance. You can do forward genetics if you don't know the gene. You clone the gene afterwards. Okay? But this is just to show you that all this population is, it ends up being grown in pots in large numbers, very carefully, as a family, and then bunch of leaf is taken from these small plants, and any mixing of DNA at this stage, you will never find the mutant in the seed packet. Often you will hear sometimes that people are saying, especially academic labs have come across that they find the mutant in the gel, but then they can't find the mutant in the packet. So this is actually, traceability is very important in this technology. So I'm now going to show you some examples. So we have long shelf life, very, very common trait, but still there's a huge interest in this trait. So this is a NOR mutant, which fruits were scored 60 days after harvest. So actually there is a NOR mutant in a Spanish variety called hanging tomato. If you Google, you will see that they hang these tomatoes like onion, garlic, and okay, and basically it is NOR mutation, 124 days LSL after harvest. Okay, this is to show you that you can do translation research in the sense that, once the melon genome was sequenced, put in public domain, we found that NOR and RIN, everything exists in melon. So, we created a population in melon, screened it, and we got long shelf life in melon. And this is a work, not mine, published in PLOS Biology, again by the French group, where I think they did ACC synthase, and they showed that there is a shelf life increase in melon. So that's the beauty, that you can take knowledge from tomato, apply for a trait in another species, and it works. Virus resistance, in a favorite topic of mine, show you the power that you can use this to create disease resistance. So potyvirus resistance, I have chosen that because we have example of that. So potyvirus is the largest genus of the plant viruses. Okay, so some of the common ones are chili, cucurbit, ZYMV, MNSV, and papaya ring spot virus as well. So a little bit of science here. Okay, so it's a RNA virus. So it has a poly A tail and a cap protein. And what happens is that this virus goes inside the plant. Imagine it's like a key. It goes inside the plant. It physically attaches itself to EI4E, okay, protein family. There are, I think, 5 in tomato and 3 in, I think, papaya and other species. So EI4E, EI4G, EI4E iso, and all that. So it's a translation initiation factor gene. So it plays an important role in initiation of translation inside the plant. So what happens is that now the virus, after it physically attaches, it is actually fooling the plant translation machinery to actually translate its RNA. Remember, it's an RNA virus. So it's now viral particles are being formed, plant is infectious. So it was shown by positional cloning by the French group that if you have a mutation in this EI4E or one of the members, you get resistant plant. And that was the basis of the project in papaya ring spot virus, because it's a potyviruses. Here, actually, I'm showing you that breeders were actually already using resistant lines without knowing what was the mutation. After this information was captured, they went back and sequenced, and they found all the potyviruses resistant line were carrying mutation in members of this gene family. In the case of pepper, what happened was the resistance broke after 3 years, and then they went back and they stacked with another, so double mutations, and they found that they were able to create resistance again. So basically, it's a key gene. If you want virus resistance in any crop, we kind of guarantee that if we have the desirable mutant, you will get resistance. So we, okay, and here another example. So this is a published paper in tomato. I borrowed the slide from Dr. Bendahaman. So he found in tomato, when he was doing tilling, he found a mutant, which was an intron splice mutant shown here. So normal gene is 5 exon. So you get a protein here. The splice mutant is defective. It makes a 3 exon protein. Normally, this should not be here. It should be, unstable, but it still is hanging around. This is a Western blot. And then Dr. Benhamer, what he did, he showed that actually you can complement the wild type. So you can complement the EI4E mutant of yeast with the wild type EI4E protein, but not the mutant one, the 3 exon one, showing that even though the proteins hang around, but the virus is unable to interact and multiply itself. And then he did an ELISA and he showed that, they're completely immune to these two viruses. And this was the basis of the papaya ring spot virus. So we created a population and we screened for mutations in this gene. And we were lucky to find a mutant which is resistant. It is being checked by 2 seed partners, one in Bangalore, one in Thailand. And we filed a joint patent on this. And just last month, our US patent has been granted. So this is something, I would say, a breakthrough, maybe discovery or contribution of our company using tilling. So other traits. So basically, as I explained to you that we mine the literature, companies come to us. We are looking at some traits which are actually specially controlled by recessive mutations. So, so these are the common traits companies ask for. Drought tolerance. Yes, there is a gene called DST in rice, drought and salt tolerance, a transcription factor. So one gene doing 2 functions. Okay, disease resistance. I explained to you that forward genetic screens you can do. At the same time, there are now actually reports of even DNA viruses going inside the plant, physically interacting, and maybe you have mutations in that, and perhaps Gemini virus resistance we can get. So we have no data on that, but I just saw a paper on Bigomo virus recently, so that would be interesting. Parthenocarpy fruits, this is of great interest in the sense that you can have fruits which set without pollination because bees population are going down. You have high heat, cold, and there are a lot of information in these areas. Also in tomato, recently a paper has been published by the French group. Increased nutrition, high lycopene or more carotenoid and all that. There are genes for that. Grain number size, in rice, many QTLs are turning out to be single genes, which is a surprise. So the central dogma, is changing because of all this new genomics technology. So there are, genes like increase the grain number, the size, the length, width, and all that. And so I don't have any data on this. So, but this is how we're working, So to summarize, in summary, so basically our services. So basically we can help you create a trait. We can help you in these various steps. So, creation of population, screening using enzyme method. Although we are switching to this one, it's much more efficient and faster than this method. But sometimes we also validate the Illumina method is confusing. So we go back and see, is it really a mutant? So that's how we double check. Phenotyping as well is getting very, very, interesting for many seed companies. People are talking about using drones and things like that. So being in India, you can use large population for phenotyping. So that's the advantage we are trying to capture. And as I said to you that, this technology can be used in all these kind of crops. We have worked on most of these crops, even medicinally, we work on Artemisia, fruit crops as papaya and things like that. So any crop you have in mind, as long as you can give us the seeds, hopefully we'll be able to help you solve your problem. And I think this is my last slide. Yeah, so thank you very much. So, so some of the projects I'm unable to show you, they have been funded by Tata Trust and Cefipra. Um, and I have a very strong collaboration with the French group because I took the license from them. So they really help and support us. And also thanks to my university, which is allowing me to um, run this company at the same time, keeping my faculty position, hedging faculty position as well. That helps. Thank you very much. Happy to answer your questions.
So thank you, Dr. Chatterjee, for the nice talk. I wish it would have gone on for a longer time where we heard more details about the different products which you've come up with. But there are a few questions which the participants have asked. I mean, a lot of them actually. I'll put across some of them to you. Otherwise, they're there in the chat box if you are already
Yeah,
logged.
I have not. Okay, so I have to stop sharing to see the chat box. Am I right?
Yeah, you can do it directly, or I can ask you the questions.
Yeah,
Yeah. Okay.
that would be better.
Okay. Okay. I'm looking at chat, but yeah, please ask me questions. I think easier.
Yeah, one of the things is that can tilling be done for polygenic traits? One of the questions.
Well, yes, you can do it in the sense that it's tedious. The process is slightly slower, not high throughput. An example I will give you is waxy wheat by tilling. They have also, by a company called Arcadia in the US, look at their website. They have also created wheat without gluten in subunits. So it took them, I think, 5 or 8 years. So what you have to do in this case is mutagenize, one by one by one by one, then cross and stack. So you can imagine the painful path, but I think it can be done. I think that's the answer I will give.
So in this respect, I have a question myself. I wanted to know as to if there are more than one mutations, some of which are when you identify the particular mutant by tilling and for the phenotypic traits, what if it segregates in the further generations?
So yeah, so what happened? So you see, like what happened is you have to understand that, let's say you don't have mutation, okay? You are crossing a wild species with a cultivated species, right? So in that case, also what you're doing, you're accumulating many unknown genes, but you are focused on your trait, okay, by eye. Here, what's happening is that we have mutations in the background, but we also have a mutation which is very specific to my trait, okay? And so what is happening is I already have the marker because I know the gene. So I cross and I use the marker to follow. What I'm doing it by crossing and back crossing is that I'm trying to make other mutations in a heterozygous state, right? So you have to follow your phenotype which you are after, let's say, long shelf life. So you can cross 3-4 times and you keep sequencing and looking at that, yes, is the line looking like the original parent which you started with? Because we are starting with, either parents of a hybrid or elite material usually, right? So this is how you do it. So in the end, you look at a plant which is looking like the original after 3-4 back crosses, but then you have the marker, and then the marker leads to the trait you are interested in. So it is really like a traditional breeding, except that you have to you have a marker in this case. So marker based back crossing. I hope I'm able to convince you. Actually, what happens is theoretically it looks, oh, it's not possible, you can't do it. This is the problem I have. And I always say, look, let's do it. I can tell you that when we are creating populations, we have got phenotypes which we never expected. And then breeder comes running saying, oh, I want this trait, at least let's maintain this plant, So I feel that, it's easier to just try it rather than saying, oh, it's not going to work. And really, this is something I'm finding with my populations actually.
There is another question from one of the participants who says that can the mutated gene be fixed by selfing?
Yeah. So well, if you can make haploid, if you are lucky, because one thing I should emphasize is that when I say I get a mutant, I don't get one mutant. Okay, I get 15 mutant or 5 mutant or 10 mutant. Out of that, you will see that maybe out of 10, maybe 3 are looking very similar to the original parent. Okay, and maybe the others are there, but they're weak and sick and all that. So we pick the ones which are very close to the original parent if we can. We call them leader alleles. And then, maybe after 1 or 2 back crosses, you can make, if you make haploidy, maybe you can fix it straight away. It is being done actually in companies. They take they make the cross, and actually they take the embryo and do embryo culture, to save time basically. That's one thing, because they're making hybrids. And the other one is that if the mutation is lethal, they actually do embryo rescue. That's how you maintain your plant. But at the same time, yes, if you can do anther culture in some species, you can do it. You can fix it.
So, another participant has asked, like we have technologies like CRISPR technologies, which are not considered as non-GMO. So, does tilling have the same risk?
Okay, no. This good question. This question is being asked by many people that is tilling non-GMO because it is GMO. You are modifying the plants. Actually, what is happening? You have to step back and think legal definition. So what what lawyers and the government regulation says. So government regulation says that if you have used agrobacterium, I am talking about European law. If you Google and see legal definition of GMOs, a method to improve plant by using agrobacterium or something like that. So if you have used agrobacterium to put your construct, it doesn't matter whether the product has the transgene or not. It is considered GM. Okay. In the case of tilling, what's happening is I am not adding anything extra from outside. I'm kind of shaking and shuffling the genes which are existing in the plant. Okay. So let's say tomato has 40,000 genes. In the case of GM project, you will add a BT gene, so it's 40,000 plus 1 gene extra. In my case, I will work on, let's say, long shelf life. It's actually 40,000 and I mutate. In the end, it's 40,000 gene. I have not added anything extra in the plant. So that's a simple way to explain that it's the legal definition you have to follow. Legally, I can tell you it's written in the European dossier that chemical mutagenesis is considered traditional method of breeding. There is no risk because look at Green Revolution wheat. They're all mutants, they're still growing, right? So they are stable. Second thing is in Vienna, in Europe, International Atomic Energy Agency, they have a plant breeding department. They are creating every year, I think, 30, 40 varieties of crops for around the, Africa and other parts of the world. Bark in our India has 40% of groundnut is actually radiation induced. So, if this tomorrow say, well, this is also GM, it's going to be a problem, So I think that way we are safe. And I'm very proud to say that it's the only legally accepted non-GMO technology,
There's another question from a participant who wants to know that recently non-coding RNAs are known to control many traits. So in such cases, would identifying mutations in genes be meaningful?
Yeah, I mean, if the non-coding RNA is having some function and you think that that function is linked to the trait of interest because of, some changes, I think, yeah, I mean, you can, with Illumina, you are scanning the genome, right? You sequence the genome and you scan using softwares and you go to that non-coding region. Yeah, I mean, absolutely. I mean, if we are screening promoters, there are now reports that if you mutate the upstream of the 5' ATG, a few sequences, you actually do, there's a Nature Biotech, so you actually do a overexpression, you know. So this means that you can, so most of the examples of tilling is down regulation, but it seems that maybe if you have a screening method, maybe you can actually even overexpress the genes, you know, by looking at certain motifs which are controlling that.
I think there is a question which Dr. Gupta has, which is related to this question. So maybe Vidya, you can put it directly.
Yeah. In fact, it was similar to what he just now said, that if there is a mutation in the upstream region of the gene which is controlling the expression of the gene, then only by using gene sequences we may lose the mutation. But if we do sequencing, uh, maybe of a those upstream regions of the gene, those mutations can be identified, and then the overexpression which is responsible for a trait alteration can be identified, you know. So
No,
sequencing has made it probably
is,
much
yeah,
better.
much high throughput, and you know we are seeing, you know, we are seeing, you know, in papers which we thought this is not possible. So I can tell you there is a gene in rice called EEL gene. It's a QTL. They did positional based mapping. It's a single gene, cytokinin oxidase. It was a science paper, and all it is doing is that when the plants are getting older, you know, the cytokinin you know, you must have, I mean, you must have, you know, you
Yep.
must remember that earlier, you know, we used to overexpress cytokinin to keep the plant greener, right? So this is the enzyme which eats the cytokinin. So if you mutate this gene, the plant stays green for another 1 week. So you have photosynthesis, and apparently 50% increase in grain number, single mutation, you know, yield. So that's why I'm saying that I think today I will say it is possible, but of course you have to do it. You know, you have to of course use logic and science behind it. You know, of course stealing has some drawbacks as well. It's not a magic bullet. But you know it's helping us to create, at least at a low cost, traits which it is delivering. You know, so I think this is what the beauty of this technology is.
I think, can I ask? One of the drawbacks, could you please list the drawbacks of this technology? That is.
So the drawback is very labor intensive. You know, you need to have a population. You have to be very careful. If you make a mistake, you have to start again. Okay. You use some crops very difficult to mutagenize and you get a good mutation frequency. Okay. You use radiation, you have deletions, and then you have problems that you get the mutant, but those mutants are unstable. You know. Okay. This is the second one, is that I can't create traits like BT cotton, insect resistance, for example. You know? I can't create traits like in polyploid very easily. You know, people are doing it, but it is not as easy as in a diploid. You know, what else? Yeah, I think, and overexpression. So yes, there are examples, but very few. Okay, so a good example is DEET1, which is a repressor of the lycopene pathway. So what happens when the tomato becomes red? It sends a signal back that I'm red, stop now lycopene. So this gene then switches off the lycopene production. People have shown by antisense and also now by chilling that if you mutate this gene, you get deep red color, because lycopene is continuously now being made. You know, so few examples of overexpression, and more examples of many of the disease resistance genes are dominant genes. Gemini virus ones. And they are the important, disease which we need to tackle, and it's not so easy with chilling. So these are some of the drawbacks.
So besides the large populations, the time required till you get the mutation, which is like a mutation with no background mutations present, what would be the approximate time frame for this?
So I basically, this is another drawback, where basically in the case of tilling, you have to cross at least 2 or 4 back crosses. So I can give an example. I think in tomato, there's a company in India called BioSeed. They have apparently released, it's a Hyderabad-based company, a long shelf life tomato. They didn't use our line. They licensed it from Arcadia, the US company. They told me that they did, I think, 4 back crosses. So after they got the line, so they got the homozygous mutant, which was long shelf life. Then they crossed it. So 4 means I think 2 years. So maybe 3 to 4 years after you found the mutant. So I think that's still not as fast as we would like to, but compared to editing, but yeah. So
There is one more question from the participant saying that is the company also working for herbicide tolerance in mustard?
Yes and no, in the sense that um, yes, I mean we have done some work in mustard, Indian mustard. At the same time, what has happened is that some of the old herbicide are actually getting kind of outdated. So like the common ones like atrazine and ALS and all that. So I mean we can do it, but if there is a new herbicide they have, we have to work together with them to create the herbicide tolerance. But the beauty of herbicide tolerance is that you can do reverse genetics if the gene is known, like ALS, or you can do forward genetics by spraying and then selecting the lines and then maybe sequencing them. So yeah.
And one participant has asked that in case of cross-pollinating plants, the commercial tilling population may get crossed to the normal population. So what will be the procedure to get around this?
Okay. No, in the case of cross-pollination, you have to self-feed, so you bag them. So the sunflower I showed you, we had to bag them. If you get cross-pollinations, it's not a problem. It's just that allele has got diluted, so you will still recover it. So the pooling strategy has to be modified. So.
I think, yeah, there are several people who want to know how large should an M2 population be for polygenic traits.
Oh dear. Very difficult to come up with a number, but I will say that if you want saturation mutagenesis, maybe aim for 100,000, perhaps. Look at the wheat paper of the vaccine gene. I think they talk about that.
I think this is about all which I could summarize from the questions which they've asked. Of course, some of the participants want to know whether whether the company conducts workshops for students and whether there are any vacancies which you have in the company.
Okay, workshops, not yet. We have been thinking about it, but it's we are a small company. It's a big effort, to have a workshop. Maybe we can think about that. Second thing is jobs at the moment, because of COVID, the activity of the company is a bit reduced. So at the moment, no, but hopefully maybe end of this year, we might, have some positions. So yeah, so send me a mail. I think it's there in my last slide. So we can discuss it directly.
Do you want to add something, Vidya?
Yeah, I think there is one interesting question by Dr. Sridhar. He says in case of tomato, the no water fruit, you have crossed with watery tomato genotype. Similarly, if salt tolerant and salt susceptible varieties, if they are the mutated genes are identified, then they can be crossed and map-based gene analysis can be done.
Yes, of course.
Yeah, that is.
Yeah, absolutely. No, so the watery less one is not my I've got a line mutant, but it was really commercialized by Noonums. If you can Google, you'll see it's called non-leaky tomato. And actually in Europe, you can get tomato salads now, like normally you cut, there's no water in it, So, and all the burgers I understand is actually using this tomato because the bun don't become soggy. Okay. And you have to also understand that, because somebody I think asked a question that um, you know, the seedless lines. How will you maintain? You know, so you have this, you have the siblings, you know, which are heterozygous, which have the seeds. So we work on that.
Okay. Yeah. And I have just as a service provider, the
Yeah.
part of your company as service provider, do you have populations ready, M2 populations for different crops, 17, 18 crops that you have worked on? Do you provide them as a service to people, the seed, M2 seed?
No, in the sense that you have to understand that the line I'm using belongs to seed companies, most of them.
Oh, so those are not produced by you for your own purpose.
So I have a project. I work on those lines, and then I give the mutants back to them. I give the box of seeds back to them, DNA back to them, and I have to declare in a letter that I have given you everything. I don't keep anything.
Yeah.
You know?
Okay. Okay.
We have this law, you know, in India about NDA and all that. So these are foreign company material.
Okay.
Okay, and now if you have for tomato, our own, we have something called M82, which is a public line which we obtained. So for academic, usually it's your line, so we don't touch your line. It's your line, it's always yours. We do not have any rights on those lines. This is how we work.
Yeah. No, but do you have your own lines created, like Arabidopsis, you know, gene bank, like that? Do you have?
Yeah, well, tomato we have only. Arabidopsis, no, I don't have. Even Arabidopsis, actually, we worked and we gave the lines back to the people. We are kind of, you know, I think people are very sensitive about their material. So we find that they don't like to give you to keep a copy and things like that. So I have to create my own. Then I have to find a line which I have complete freedom to operate. You know, it's to start this what you are saying. You know,
yeah,
it's possible, but so far last 14 years we have not done that. We have been working with customers' material, but tomato is our model system, so we have been working on tomato.
Okay, yeah.
One more question has just come in. That is, would this type of technology, what would be the cost per seed as compared to the BT technology? If you produce a similar type of product using tilling, the transgenic technology, what would be the cost per seed?
Well, that's
whatever.
Okay. No, so you know, I can tell you like BT technology actually now, of course, the price is kind of subsidized. I think it's 800 rupees or something per packet, right? So, so I don't know. Depends on the crop species and all that. So, papaya ring spot virus, I am told that the cost of papaya seed, the tolerant one today, is about 3 to 4 lakhs rupees a kg. This variety called Red Lady, which comes from
Taiwan.
So it is not non-GM, although people are saying it is. But so I'm hoping that, you know, if I start my line, the cost of that will be maybe an additional premium of 5% perhaps. So it's very difficult. You know, it's an interesting question in the sense that it all depends on. Of course, it will be cheaper than cheaper to develop the trait compared to a GM trait because of the regulatory cost. And so but the price is fixed by the seed companies. So I'm hoping that logically it should be less costlier than a GM trait. So I think this could be the answer I can give.
Okay. And one of the persons I think working in a university, he has asked that if they want to run a project on tilling, would your company be willing to provide tilling facility, and the crossing and preparation of the populations can be done at the university?
Yeah, I mean, we have worked like that with some companies also. Yeah, it's possible. Yeah, yeah.
What would be the cost involved for?
The cost, it depends. You know, it depends on the crop cycle. You know, if it's papaya, it will be expensive, 12 months. If it is tomato, it's 6 months. Then the you know, how many genes to till, and all that. So he could maybe send me a mail privately and directly, and maybe we can discuss that.
And is the price any different for a university person and or from the public domain government service person as compared to the companies, other companies, seed companies?
Yeah, so definitely we try to, we understand the government limitation and also Indian companies. So there's a difference, definitely, yes, of course.
Okay. I think we have covered most of the questions and maybe we can end the session now. I thank you very much, Professor Chatterjee, for a very nice talk. And if there are any questions which the participants want to put up to you, maybe we can ask them to mail you directly. Is that okay?
Yeah, absolutely.
Okay.
Yeah.
So then we can end the session for today. And I request Prachi to end the session in case she has any announcements.
No, just one announcement that tomorrow the session is at 10 o'clock as usual. And the same link would be working, which is
Yes,
all.
yes, yes.
Okay, see
Yeah.
you all at 10 o'clock tomorrow, sharp.
Right.
Thank you
Okay.
so much.
Thank you, Manash.
Yeah, I would like to thank you to the organizers. Thank you to all the participants. Thank you very much. And please contact me directly if you have any questions. Happy to answer them.
Okay.
Bye-bye. Have a lovely day.
Bye. Thank you.